Hello and welcome to the ABE Labs 1 to 4 refresher video. My name is Anna Wedderburn. I'm the current ABE Ireland Programme Coordinator, and I'm just going to do a quick refresher of ABE Labs 1 to 4. So what we're going to go through is we're going to go through the theory and content and setup of ABE Ireland Labs 1 to 4. We'll do a quick review of the Lab Exchange platform and the teaching material and also the programme logistics. So just to refresh your memory very quickly, we've got 4 labs. So there's lab 1, which is micropipetting. There's lab 2, which is DNA profiling. Lab 3, which is DNA digest. And lab 4, which is DNA ligation. And we're going to be going through all 4 of those today. So you'll be aware of this already because you'll have got this video through Lab Exchange. But just a reminder that all of the teaching materials that you need are available to you on Lab Exchange. This includes teacher manuals, sample timetable for classes, worksheets for your students, video setups for the equipment, and videos of career talks, all that sort of stuff. And just so you know, before we get going as well, this is the size of the kit. So this is a picture of me beside the kit. So I'm 5'4"-ish, slightly under, and you can see that there's 5 boxes, sometimes a small 6th one as well, and so it takes up quite a lot of space. So bring an empty car when you're going to collect the kit. And just so you know, these boxes, they contain all the equipment and consumables you need. The only thing we don't provide is a water bath, which is needed for lab 3. Okay, so the first lab is all about micropipetting. So students learn how to use a micropipette. So this is a picture of one here. The pipettes that we use are P20s. So this means that they pick up 2 microliters up to 20 microliters. So a microliter is unit of measurement used in a lab. So 1 microliter is 1,000th of a mil, and a millionth of a liter. And just to give you an idea of what that kind of looks like, a raindrop contains approximately 34 microliters. So in terms of actually holding a micropipette, I'm sorry I don't have one with me today, so I can't actually show you. But this diagram shows it pretty well. What I tell people do who haven't used a micropipette before, is I tell them to put out their hand as if they're giving a thumbs up, and then the kind of like hook part of the micropipette like hooks over the front of their fingers, and that's how to hold it. So you've got the plunger button on the top. That plunger button also twists, and that's how you adjust the volume of it. You've got the tip ejector, which you use to get the disposable tip off the bottom of the pipette. You've got a digital volume indicator, the shaft, and disposable tip. And just so you know, there's a really good simulation of this on Lab Exchange that I'd really recommend using when you're going through it with your students. It's really helpful. So in order to set the pipette to the right number, basically what you've got to do is look at the numbers on the side. So this first one here on the left, that's 20 microliters. So the first 2 numbers, they're kind of like the whole numbers, and then the third number is the decimal point. Sometimes the decimal point is red. It's really helpful when that is the case. It's not the case for all of our pipettes though, unfortunately. So the first 2 whole numbers, and then the third one is a decimal point. So the first number we're looking at here is 20.0, the second one is 12.4, third is 5.5, and then the final one is 2.0. And then, like I mentioned earlier, the plunger button on the top, it twists. So if you twist it to the left, the volume increases, and if you twist it to the right, the volume decreases. It's really important to let your students know to never twist it never put it below 2 microliters, and never put it above 20 microliters, because it will break. And they're at least €200 each. So another thing to note about the micropipettes when you're using them is there are 2 stops on the plunger button. So you put your thumb down, you kind of feel resistance, that's the first stop, and then you can push through that to the second stop. So when we are using micropipette, what we do is when we're picking up liquid, we go to the first stop. So you go to the first stop, pick up the liquid, bring your thumb up kind of slowly, and then move to wherever you're wanting to move it to. And then for that, when you're expelling it, you go all the way to the second stop, and that just makes sure that all the liquid actually gets kind of kicked out of the pipette. So this is a diagram of the way to do it. So step 0 is putting the disposable tip on your pipette. Step 1 is going down to the first stop, with your thumb. Then you lower the pipette into the liquid, slowly draw it up with your thumb, move over to wherever you're wanting to move it, and then you go all the way down to the second stop. Then remove the pipette and eject. So that's how to use a micropipette. So this is the kind of the setup for lab 1. So we give you a micropipette and a box of disposable pipette tips. We give you 1 pipette between 2 students, and then there's a beaker there. You can see we use that for waste pipette tips. So once they're done, they can eject them into the beaker. And then some different colored dyes, which I'll go through now as well. And then the other things you see there is a pipette template sheet, practice gel electrophoresis gel, and then there's kind of like an additional activity we've put in the last couple of years called sun catcher activity. So it's a 96-well plate, if any of you have ever used that before. So the 2 kind of original activities that were involved in lab 1 were this practice pipette sheet, which you can see on the right. So basically what students have to do is just set their pipette to the right volume, pick up the right volume of the red dye is what we use here, and then just like pipette above that black dot and go all the way down to the second stop. So if they kind of pick up the right amount of liquid, if they have steady hands, and if they've done all the first stops and second stops correctly, the dye should stay within the circle around that black dot. So as you can see in this example, that was not the case. So that was someone who hadn't quite managed it for the first one, but then they got progressively better, so went down. So the first one they've got to do is 20 microliters, second one is 15, 10, 5, and then 2. My advice for this one would be hands can be quite shaky, I guess. So what I do is I put both my elbows onto the table or the lab bench, and then I kind of like help, so say this is my pipette, I help guide it in with my other hand, like that, so then I can pipette in like that. And then the other thing to note here is not to put the pipette tip like directly onto the black dot, because that'll actually just like block the liquid from getting out. So that's the first activity. The other thing we do in lab 1 is we give you some practice gels. So this kind of comes into play in labs 2, 3, and 4, where we'll be doing gel electrophoresis. So in lab 1, we just get them used to the idea of pipetting into an agarose gel, because they're kind of tricky. Like the wells, you can see the wells here. The rectangles, they can be quite tricky to see because everything is invisible. Like the gel is invisible, the wells are invisible. It's all a bit invisible. So what we get them to do here is just 8 microliters into the practice gel. They're just pipetting in. And what I recommend doing here as well is like just to get an idea of what the wells are like, first of all, is just pick up one of the disposable tips in your hand and kind of like wiggle that around within the well, so you can kind of have an idea yourself what the well feels like before, and your students before going in with a pipette. And again, I use my 2 elbows and kind of like guide it in with my second hand there. And it generally tends to be 1 practice gel between 4 students for this. Then the activity that we have kind of added in the last couple of years is to lab 1 is this sun catcher activity. So basically we're getting the students to make pictures. There are 5 different pictures, A to E. So we give you a set of coordinates, we give you a 96-well plate, and we give you some colored dyes. And what the students have to do is, so say for this one, for example, you can see that the coordinate says red dye, C11, D10, E9, E11, E12, F10, G11. So red dye goes into each of those wells, and you put 40 microliters in. So it will be 40 microliters of red dye into those wells. So 20 microliters twice. So these are what some of the pictures look like. So there's a butterfly, a car, a fish. There's also a rocket and a flower. So that is lab 1 complete. Lab 2 is all focused on its DNA profiling using gel electrophoresis. So again, we'll be building on the skills that we just learned in lab 1. So in this lab, basically, we have said that there is a crime scene. There was a jewelry robbery, and we've got DNA from the crime scene and DNA from our 1, 2, 3, 4, 5, 6 suspects. And the students just need to load the DNA into the gel electrophoresis tank and then run it and like match the crime scene up with the suspect that did it. I'll go through the results of that now. So basically, you get given these samples in a box like this. And there'll also be a DNA ladder for you as well. And then you've got to set up the gel electrophoresis tank. And again, just so you know, there's a really good lab simulation on Lab Exchange that walks you and your students through gel electrophoresis and what it is, all that good stuff. It's really, really good. So this is the gel electrophoresis tank that we give you. It comes in a box like this. You take them all out of the box. You'll see you've got a orange kind of hood that goes on top. You've got the plug to plug it in, and then you've got this little tank where all the buffer and the gel actually sits in. So the first thing to do is to put that tank or that tank into the tank. So you pick it up, pop it in. It should make a little click sound. It's important to put the minus, the negative kind of stuff up to. Can I get a pointer? I can. So the minus side signals in this conical. The next step then is to get your gel, your profiling gel. So your profiling gel is slightly different to your practice gel in that there's only one line of wells. So what you do is you then pop your profiling gel into the little tank and make sure that all the wells actually get filled up with the buffer, because sometimes when you put it in, the wells they stay with no liquid in them. So make sure the liquid actually gets into those wells. And then top tip is to make sure that you actually put the light on when all that's in, so that it really just makes seeing the wells so much easier for your students, so that they can then pipette in the various samples. Again, I use my 2 elbows and kind of like guide in for this one. And it's really important to change the pipette tips between each one, because otherwise they'll all get contaminated. So then all you do is you pop on the orange hood on top of that and hit the on button. Once the on button is hit, you'll see a little green light pops on here. If that green light is flashing, it means that there's too much TAE buffer in the tank and you need to remove some of that. One of those like dropper pipettes is really good for doing that. Otherwise, you should see this green light just like constantly on, just on. And if you look inside here, you'll see some small bubbles coming up from the little metal bar that's up the top here. And then you can just leave that running. What these people have done is the people who created these tanks, is they've put in a little hole at the top. It's not a hole, but like a viewing thing here. So what you can do is you can actually like put 2 pipette tip boxes beside it, balance the phone in it, and do a time lapse if you want. These machines are totally safe. They are made specifically for secondary school students. So there's no way that they can electrocute themselves, because the hood, like the orange hood, needs to be on for the electricity to actually run through it. And there's no ethidium bromide or anything like that, which is a carcinogen, which is what you normally need to run gel electrophoresis. But there's none of that in this. Everything is totally safe. So you run that. Once it's on, you run it for 20 minutes. And this is what the results tend to look like. So you've got your DNA ladder here. You got your crime scene. So that's your crime scene there. And then we've got our suspects A, B, C, D, and E. And sometimes there's an F as well. So here you can see that the crime scene matches up with C. So that would be who did it. These days, sorry, I think this must be an old photo, but these days it tends to be suspect D who did it. But yeah, so you can see for yourself which ones match up. So for this one, it's the crime scene is C. So. And then just so you know, you probably know this already, but DNA is negatively charged. So that's why it kind of like goes down towards the cathode with the electricity all running through it. And then because the agarose gel acts like a sieve, the smaller pieces actually move through it faster because the big pieces kind of get more stuck along the way. Yeah, so that's that. And then I mentioned DNA ladder there, but I didn't actually really explain what it is. It's basically a solution of known DNA bands that we know the sizes of. So when you get given a DNA ladder, you also get given a map like this. So you know that the first band is always 10,000 base pairs. The second one is always 8,000 base pairs, all the way down to 250 base pairs. So we run them in this case just kind of to act like a control almost, so that we can see that yes, the DNA has worked, or the gel has run. So that's what the DNA ladder is. Okay. So 1 second. So that's lab 2, all done. It's a pretty simple one. It's just gel electrophoresis. It's just loading the samples. That's it. So, and all of this information, like kind of like summaries of all this information, will be given to you on like cheat sheets that will be given to you in your kit loan. And we also have this available on Lab Exchange as well. So in terms of labs 3 and 4, I'm going to just give you a bit of background information that you need before continuing on with them. So first thing we need to learn about is something called restriction enzymes. So these are enzymes that are able to cut up DNA. So they're essentially a molecular DNA scissors. So the way they work is that they're programmed to find and cut specific DNA sequences. So this restriction enzyme here would be floating around searching through DNA until it found this sequence on the screen. And once it finds it, it basically gets to work and cuts it up. So that's a restriction enzyme, basically cuts up DNA. The next thing you need to learn about is something called ligase. So this is an enzyme that sticks broken DNA strands back together. So it's basically a molecular glue. You basically just stick it in and it sticks the DNA back together. And just so you know, ligase is naturally used in DNA replication and repair, but we also use it in the lab. And sorry, something that I just forgot to tell you here about restriction enzymes is that restriction enzymes, they were actually first discovered in bacteria. So bacteria use them to defend themselves against viruses. So when the virus injects its DNA into bacteria, the bacteria's own restriction enzymes would cut up the DNA of the virus to prevent it from replicating. So that's where restriction enzymes originate from as a bacterial mechanism of defense, but we use it in the labs now as well. So restriction enzymes cut up DNA, ligase sticks DNA back together. And then the last piece of information we need for labs 3 and 4 is on bacterial plasmids. So bacteria have 2 types of DNA in them. They've got chromosomal DNA and they've got plasmid DNA. So chromosomal DNA, it carries all the information the bacteria needs for growth, development, and reproduction. So all the genes it needs to just exist. Plasmid DNA, however, it's kind of like a little loop of DNA, and it contains extra genes that aren't necessary for the organism's existence, but they do make it stronger and more likely to survive. So it could be genes for like antibiotic resistance or a stronger cell wall. So plasmids are really cool because they can transfer from one bacteria to another to kind of help their friends also become resistant to antibiotics or to have a stronger cell wall. And because of this, plasmids are really, really stable, meaning that they don't fall apart or degrade easily. So due to their stability, they can be easily used in labs by scientists. And one thing that scientists do with plasmids in a lab is that they make insulin for people with diabetes. So what they do is they get their plasmid and they cut it open with a restriction enzyme, and then they put in the gene for insulin. They glue the plasmid back together again using ligase. And then they put it into the plasmid into bacteria. The bacteria then make insulin themselves, and then which is then purified and given to people with diabetes. which is great because beforehand they actually had to use to make insulin in pigs. So these days it's just made in bacteria. The pigs are safe. So these are the techniques that are used basically that we do in labs 3 and 4. So that's kind of like the real world application of it. Okay, so the plasmid that we use in labs 3 and 4 is 6,800 base pairs long, I guess, but in a circle. So this is it here. So you've got 6,800 base pairs, and it can be cut by 3 different restriction enzymes in 3 different places. So you've got restriction enzyme A cuts it here, restriction enzyme B cuts it here, and restriction enzyme C cuts it here. So A is at 0 base pairs, B is at 1,000 base pairs, and C is at 3,300 base pairs. Great. Okay, so that's all the theory that you need to know for labs 3 and 4. In terms of actually getting the kit to you, we give you for labs 3, there are 4 different tubes basically, plus a DNA ladder. So you've got 3 DNA, 3 0, 3 A, and 3 ABC. And we used to give it out in boxes, but I think these days we're actually giving it out in little bags. So you'll get a little bag full of 3 DNA, a little bag full of 3 0, a little bag full of 3 A, a little bag full of 3 ABC, and then also a bag of 3 DNA, or sorry, DNA ladder. So these are the 4 tubes that we're essentially using in lab 3. So lab 3, just a reminder again for you, is DNA digest. So that's the molecular DNA scissors. So we're cutting up DNA. So we're given, as I just mentioned, we're given 3 DNA, 3 0, 3 A, and 3 ABC. And this is done in groups of 3 with your students. So these are the instructions. This is what needs to be done in the practical. So you got given your tube of lab 3 DNA. And then so lab 3 DNA is just your plasmid. So our 6,800 base pair plasmid. So that's what's in tube 3 DNA. Okay. So what we're going to do then is we're going to put 7 microliters of 3 DNA into each of the other tubes, changing the tip each time. So you do 7 microliters of lab 3 DNA into 3 0, change tip. 7 microliters of lab 3 DNA into 3A, change tip. 7 microliters of lab 3 DNA into 3 ABC. So one student each would do each of those things. So what's happening here is 3 0, it contains no restriction enzymes. So our plasmid should just be staying the same, really. 3A, it contains restriction enzyme A only, so it should get cut up here. And then 3ABC contains restriction enzymes A, B, and C, so it should get cut at A, B, and C. And then what you're going to do is you're going to put it in a water bath at 37 degrees for 5 minutes. So that is what lab 3 is. And then you run it on a gel electrophoresis DNA gel agarose gel in a gel electrophoresis tank to see the results. So that's what lab 3 is, okay? And what I always recommend doing is setting up the tubes in the tube racks that we give you the same way as the instructions are written out, basically. So you'll see here that like the way I've set out these tubes matches the instructions, and that just really helps get your mind around it. So 3 DNA, 3 DNA, 3 0, 3 0, 3A, 3A, 3 ABC, 3 ABC. Correct. Okay. And then we also give you just these little floaters as well to float them in the water bath. So once they've done that, we give you a profiling gel. So it's 2 groups per profiling gel. So you'd have your DNA ladder here. So there's just 1 DNA ladder for 2 groups. Then you'd have your group 1. So they'd put in their 3 0, their 3A, and their 3 ABC. Then leave a gap. And then your group 2 will put in their 3 0, their 3A, and their 3 ABC. So that is lab 3. But we haven't gone through the results yet. That's what we'll do now. So what I always like to do is I like to get people to guess what the results will look like. So we should be able to do this because we know where the restriction enzymes are going to cut up the plasmid. We know the size of the plasmid. We know all of that sort of stuff. So For 3-0, what do we expect to see? Just the plasmid really, because nothing's getting cut. For 3A, it's just going to get cut at A. So what we'd expect to see is actually this plasmid, which is a circle just opening up. So you just expect to see just one line. And that one line would be 6,800 base pairs in length. For 3ABC, this plasmid is going to get cut at A, B, and C. So we'd expect to see 3 lines, and we can predict what size those bands will be as well. Because this is cut at 0, this is cut at 1,000. So the first line will be 1,000 base pairs. This is cut at 1,000, this is cut at 2,300. So this line that we see next would be 2,300. And then we'd also get the to see the third line, which goes from C to A. And I can't remember off the top of my head, I think I've got it written on the next slide, so we'll get to that. So yeah, so this is basically what we're seeing. So we're going to have our plasmid uncut. So that's the 3-0 basically. Then we've got the one that's just cut with A. So that's why we just get one band there. And then we've got 3 bands here. So we've got those 3 there. So we've got the 1,000 base pair, which I said first, that's between A and B. Then we've got the 2,300 base pairs line, which is here. And that's between B and C. And then we've got 3,500 base pairs here. And that's between C and A. I hope that makes sense to you. So the reason 1,000 is down here is because, as we said earlier, the smaller pieces, they travel through the gel faster than the bigger pieces. One thing you will notice is that the plasmid uncut is kind of like a bit of a blur. So that's the stuff that would be in the tube 3-0. And the reason for this is the plasmids, basically they come in lots of different kind of forms. So you get like a supercoiled plasmid, which is all like really, really, really folded up in itself, and that travels really far. So you kind of see that part like down here. Then you've got a nicked plasmid where like one of the bands, because DNA is double stranded, so one of the strands in the plasmid might be open. So it's just a bit more open than the supercoiled one. So then you get something that's a bit higher than supercoiled, and then sometimes they just like spontaneously break open entirely as well. So you get some linear ones as well up here. So that's why you've kind of got this big blur for the plasmid uncut. Okay, so that is lab 3. Then lab 4. This is DNA ligation. So this is sticking DNA back together again. So again, we used to give these in boxes, but now it'll be given to you in just bags. I think it makes it easier for you. So the kind of 1, 2, 3, 4 different things plus DNA ladder that you get is ligase buffer, uncut DNA, and cut DNA. So you'll get those in lots of nice different separate bags, and they'll all be labeled that they're lab 4. So again, this is done in groups of 3 between your students. So they get given each 1 tube of uncut, 2 tubes of cut DNA, 2 tubes of ligase buffer, and 1 tube of ligase. So earlier when I was explaining what ligase was, I left out the fact that ligase can't work alone. It needs energy to stick DNA pieces together. So this energy comes in the form of something called ATP. So ligase buffer is just a solution that contains ATP. So basically the ligase uses the ATP in ligase buffer to stick DNA together. So let's have a look inside each tube again. So we've got our lab 4 uncut DNA. So again, this is just our uncut plasmid. So you'll see up there in the corner, kind of, it's just our normal plasmid. Then we're giving you 2 tubes of cut DNA. So these are cut with restriction enzymes A and B. So A and B. So you're kind of expecting there to be 2 lines here. So what we're going to do is the first tube, you just leave it, do nothing with it, just let it sit there. And that's the uncut DNA. To the second kind of line that we're doing, so you've got your cut DNA, and we're adding that to ligase buffer. So you've got your cut DNA here, so it's the 2 lines, and you're adding it to your ligase buffer, which is the ATP. So this is essentially acting as our negative control, so we don't expect anything to happen here. And then down at the bottom, we've got our cut DNA plus the ligase buffer, or the ATP, plus the ligase. And then what we're going to do, so once all those things have been added together, you're just going to leave it for 10 minutes. So you got your uncut DNA, leave it alone, just leave it 10 minutes by itself at room temperature. You got your cut DNA, so you're going to add 17 microliters of this cut DNA to the ligase buffer, and then just leave that for 10 minutes. And then for this final one, you're going to do 17 microliters of the cut DNA into your ligase buffer. So there's 2 microliters of ligase buffer in there. So then you've got your 17 microliters of cut DNA and 2 microliters of ligase buffer. So that's 19 microliters in total. So you're going to move all of those 19 microliters over to the final ligase tube. So again, I really recommend putting out the tubes in the same way as the instructions. So you've got your uncut, matching your uncut, your cut, ligase buffer, cut, ligase buffer, ligase. Great. And something that I should have said for lab 3 as well is that before doing any of lab 3 or lab 4 labs, centrifuge all these tubes, because we're working with really small volumes here. So like 2 microliters of ligase buffer, like that's absolutely teeny tiny. So you need to centrifuge them in the little centrifuge they send you, and that just makes sure that all the liquid is actually down at the bottom. So what you also want to make sure is that like your students are also adding everything down at the bottom, so that everything is actually mixing with each other. Just make sure everything's down at the bottom. Great. Okay, so you add all those things, you leave it at room temperature for 10 minutes, and that lets all the ligation happen. And then what you do is you put it in a profiling gel and do gel electrophoresis on it again. So again, we do the DNA ladder first. So that's 1 between 2. Then you got group 1 can do their uncut DNA, their ligase buffer, and their ligase. So that's one group. Leave a gap, and then group 2 can do their ones. So again, what we're going to do is we're going to kind of predict what students think they might see. So for the uncut DNA, that's just going to be the plasmid. For the cut DNA with the ligase buffer, we'd expect to see 2 lines there, because it's the plasmid that's been cut in 2 places, but it's not getting stuck back together again. And then for the final one, we'd expect to see it stuck together again. So this is what the results look like. So you've got your DNA ladder here. We've got our uncut DNA there, so that's our plasmid. We've got our cut DNA, which was cut with restriction enzymes A and B, so we've got 2 lines. So that's our cut DNA. Our negative control, the one that was just the cut DNA with the ligase buffer. And then our final sample, which was the cut DNA and the ligase buffer and the ligase, we get this big kind of blob up here. Now, what some people predict for this one, what they predict that they'd see would be the exact same as the uncut DNA. The reason we don't see that, and the reason we see a big blob instead, is because the ligase doesn't know that we just want the big piece and the small piece together, and that's it. So what it ends up doing is it actually ends up sticking lots of these pieces together. So you end up getting like this big kind of plasmid and this big jumble of DNA, as opposed to and like really long lines of DNA as well. And so they're just like a big mess of DNA, basically, because it didn't know that we wanted it just to stick those 2 pieces back together again. So that's why we get the big blob up there. Okay, so then I hope this part isn't confusing, but basically, as I said, for labs 3, you kind of have 2 groups of lab 3 running on one profiling gel, and then for labs 4, you can have 2 groups doing lab 4 and running on one profiling gel. But if you want, I know that one teacher, what they do is they get half of the class to do lab 3 and the other half of the class to do lab 4, and then she gets them to come together as one group. So then what you get for that one group is you get DNA ladder, you get the half that we're doing lab 3 running it there, and the half that we're doing lab 4 running it there. So basically you can kind of mix and match however way you want. Okay, so those are the 4 labs. So that's the theory behind them and kind of how to actually run them as well. You do get all the PowerPoint slides on Lab Exchange that you can use and update yourself as well. So next I'm going to talk about cleaning up and returning. because this is a really important part of the kit loan, especially if you are loaning kit from one of our remote centres, because the way you leave the kit will be like, we won't be there in between. Like the way you leave it will be how the next teacher will pick it up. So in terms of tips, all the used tips that you've used during the classes, they get the students empty. Sorry, God, I'm absolutely gone. So when the students are using the tips, they put them into the beakers. At the end of the class, empty all these beakers into this tip waste container that we give you in your kit. And then when you get back to the remote centre, so if you're in Laois, Monaghan or Drumcondra, there will be a metal container like this. So then empty this big tip waste container into this even bigger tip waste container, basically. It's also really important to fill up your disposable tip, the tip boxes. So we give you some refill tips, just put new ones back into the tip boxes so that they are full and ready to go for the next teacher as well. In terms of all the gels that we give you 2 bags for the gels. So there's used gels, no trays, because the gels, they all come in these like kind of gel trays. So slide the gels off them, put them into one bag, and then put those gel trays into another bag. So it says gel trays. Just make sure they're separate in 2 separate bags. There's also a bag that says used colors. So that is for all the food coloring things that you used in lab 1. They just go in there because we reuse them. In terms of the TAE that you used, we give you a funnel and a TAE buffer waste bottle. So just pour it all back in there. We take all that back. We give you yellow bags. So this is where you put all used gloves, all the used DNA samples. So all the samples from labs 2, 3, and 4, they all go into the yellow bin bag if you use them. And then also the bag of gels will also pop in there as well. There's something else there, sorry. In terms of packing back up the kit yourself, we give you 4 big boxes. They have lid inventories on them, so just put things back in the right box. So basically, box 1, you've got your pipette tip boxes, and your tube racks. Box 2 is all your gel electrophoresis tanks. Box 3 is all that stuff. So you've got your centrifuge, your 96-well plates, the beakers, the tip waste containers, the large tip waste container, the conical flask, and the funnel. And then box 4, you've got all your gloves, TAE bottles, basically all that. So anything that you don't use, keep it cold because we can reuse it. So if you had samples from labs 2, 3, or 4 that you didn't use, just keep them cold and we'll reuse them. So if you're returning them to Laois or Monaghan, pop them back in the fridge or freezer. So in terms of where we are, you probably already know this, but we've got 4 locations. So we're in UCD, Drumcondra Education Centre, Laois Education Centre, and Monaghan Education Centre. In terms of booking the kits yourself, we send out an email at the end of the academic year that lets you book up to Halloween of the next year. And then at the end of September, once we've done all our new teacher training workshops, we open up the kit booking form for the remainder of the year. So if you're watching this, you're already on our mailing list, so you'll get emails about this. So don't worry about it too much. Just a reminder that this is the size of the kit. So you got 4 boxes, 1 small box, 1 polystyrene box, which contains all your cold bits, and each contains all the is there. Put in a student's name, the date, and your own signature, and I just take whatever labs that they've done. And then just a reminder to you that we do deep dive topics. So we do a lab 5, which is PCR. We do a lab 6, which is bioinformatics. So just keep an eye out on all the emails for that, because we'll always advertise it through our newsletter. We also are involved in JCT activities, although they're now IJA, so we'll see what happens there. And we can also provide guest speakers to your classroom as well. So all you have to do there is email us at abe-ireland or abe-ireland@ucd.ie asking for a guest speaker, and we can arrange for a researcher to come and visit your class and talk to your class. So that is it. Thank you very much.